you'll get your biolog plates back and look at those just visually. And then we'll get those put through the plate reader. So it'll be like probably a 72-hour incubation read of when they'll get assessed with the plate reader. Hey, with your water samples, you were doing those Durham fermentation tubes, right? They pop yellow? Did they go from red to yellow? Okay. So then you'll be doing, you probably on Friday streaked onto EMB, right? So then today you'll look at those results. And then the other thing you have to do today is set up 2 experiments. They'll be quick to set up, but Duncan needs to spend some time talking to you about them. But we're going to do start 2 experiments that are kind of running through till next Monday, essentially. And so you're going to hunt for nitrogen fixing bacteria. And then you're going to sample yourself. You're going to look at yourself as a habitat and do some sampling. And then on Friday, you'll work with what you grew from your body. So fun couple of exercises there. All right. Let's see, where am I? I think we left off. We're covering methods. How do you assess microbes once you get your samples back in the lab? And I was looking at this idea of if you want to count, like if you want to know population estimates of your sample, then a flow cytometer is the way to go. Much, much quicker and way more accurate than having a technician look through a microscope and do direct counts on a microscope, that fancy microscope slide, the hemocytometer. So flow cytometer is really good. You can also get super fancy with it and stain them and sort them. And so it has a lot of applications, not just giving you population sizes. You can get some other information here too. One that I've never seen happen, I've never seen this, but laser tweezers is a thing where you can pinpoint really slow growing bacteria from a mixed culture experiment that you're running. And so apparently what they do is they shoot laser beams and they're able to like trap bacteria in the beam and capture them in a mixed sample. Optically trapped cell. That's pretty fancy. Who thinks up these things? That's what I want to know. Pretty crazy. So this is, I could see its benefit if you're using, so for example, a lot of microbial ecology experiments, oh, I don't know if I have that in here. You use a chemostat. I don't know if I put that in there. I should. Chemostat, which is a piece of equipment. It can be as small as something that you put on a bench top and about the size of a coffee maker, or it can get quite big. Depends on what you're doing with it. But essentially it is a system that is hooks up to hoses and allows you to pump in fresh nutrients. It allows you to siphon off used nutrients and waste. And it allows you to keep your bacteria or whatever, your microbes in a steady state of growth so that they're not going to crash and die swimming around in their own waste. Okay. Your gastrointestinal tract is a good example of a chemostat. You're a walking chemostat because you take in fluids and nutrients, you get rid of waste, and that way you keep the microbes in your gastrointestinal tract happy and fairly stable. And so I could see the benefit of taking samples from your chemostat and trying to isolate some of the minor populations in that sample. But I'm guessing this is a fair bit of money, so you'd have to get a good grant for that one. Epifluorescence has been around for a long time, but it's still heavily used. This is something that has really flourished over the years in terms of your options available and in terms of stains. I'm just listing out a couple here. Acridine orange is still really commonly used, Dappy and Fitzy. So there's all these different stains that you could buy from companies, and they'll tell you what it will allow you to do. And another one is benzamide stain, but it's been, historically, it's always been a challenge to do epifluorescence with soil samples, but now they've developed enough stains out there where you can start to tell the difference between the soil background and the cells will pop in contrast to that. And so you're able to get some good images even with a soil sample. And then there's fluorescent antibody techniques that are still heavily used. And so antibodies, that means you can tag a fluorescence dye onto an antibody that is going to match up with some microbe you're interested in that population, for example. So if, for me, if I took a soil sample and said, I want to know how many Streptomyces scabies species are here, I could develop an antibody specific to that bacteria and tag it with a fluorescent stain, and then search for a hit, positive fluorescence for that. You got to be very careful with your processing of the sample because you have to be the background issue where the background doesn't fade back. Sometimes stains too bright. So sometimes that can be a problem. So you have to sometimes troubleshoot that. But another technique you could do is a gene probe. That should probably be its own bullet point, not a sub bullet point. But a fluorescent gene probe is similar to the antibody idea where you can ask, if you take a sample from a habitat and say, is this gene in this population? Is it there or not? So maybe you're looking for, maybe you go to a contaminated site that's contaminated with benzene, and then you ask the question, are there microbes in this soil sample, for example, that have the gene to break down benzene? If you know what that gene is, you can then do a technique that you could actually identify the microbes that have that specific gene in that population. So there's ways to get at this with staining and microscopy, including confocal. You can do confocal microscopy. We have a confocal microscope in our department that works really well for these types of things. Here's some images. So Dappy is the figure A and Acridine orange is figure B. And you can see some differences in color. Figure C, that was a sample taken from Puget Sound. And so they were hunting for certain bacteria there. And so you can see if you do it right, if you have a good technique, it really does give a vibrant contrast of the microbes to their background. And so that can be very effective. This idea of gene probing is a protocol that's pretty well established. And if you follow it closely, you usually can get it to work without too much troubleshooting. If you have some DNA isolated from your sample, and then you put it onto a special membrane, and then you process it where you denature it, so you can get it single-stranded. And then you add a stretch of DNA that will bind to it with your probe on there. And then when you're done, you can see if there was any binding taking place. And then you can get it to fluoresce as well if you want to tag it with a fluorescent dye. So these techniques some of these techniques have been around for quite a while. Some are fairly new and they continue to develop, and the protocols get better and better in terms of you have to do less troubleshooting. Here's a comparison just showing you how In this case, they looked at epifluorescent counts, direct counts under a microscope versus viable count. So anytime you hear viable count, we're thinking about getting that sample spread out onto a plate and then incubating that plate and seeing how many colonies show up after incubation. So viable, you're only counting the viable microbes, the ones that grew up over that incubation period. So direct versus viable, and they had two samples, soil and marine water. And in this case, you can see that direct count wins in terms of getting a more accurate estimate of population. Which I'm not terribly surprised, because viable count, you would have to do, when you spread something out on an agar plate, what recipe you provide makes a big difference on who's going to grow on that plate. Not all microbes like nutrient agar or tryptic soy agar. And so if you really wanted to do a viable count, a really robust, you'd have like 10 different agar types, and your experimental cost would go up quite a bit. And so you're kind of limited by cost. Agar is expensive. In fact, I got yelled at that my classes are a little too expensive lately. It's like, we're doing experiments, come on. So anyways, agar is expensive. And so if you do a viable count, you are always hemmed in by that reality. And so direct counts usually are a better way to go, especially if you have a good epifluorescent technique where you can do this. So if your question is, what's the population size? Flow cytometer or epifluorescence direct counting would be a good strategy. Most people do epifluorescence because a lot of people, they just don't have access to a flow cytometer. Okay. There's this thing called a reporter genes. And I guess they named it, well, they report whether or not your microbe is in that sample. So it's a way to track microbes, whether you put them there to begin with, or you're just hunting for them, wondering if they're there to begin with. And so I've done some experiments with LAC-Z gene. So this is a gene that you can have in your microbe. You can put it into your microbe, and then so for example, if I wanted to say, I wonder if the streptomyces I'm working with would be a good biocontrol agent. One thing about being a good biocontrol agent is that when you put it out into the soil system, it persists. It just doesn't immediately die off. So then I might put the LAC-Z gene into it and put it out into the environment, let a growing season go, and then sample over time and say, is the microbe still there? Is it still persisting? Because that's a hallmark of a good biocontrol agent. So then I would take those samples, go back to the lab, and it's really nice because you do some viable spread plating, viable plate counts. And when the colonies show up on the plates, because of the LAC-Z gene, you've added in this XGAL chemical into the agar. And when the bacteria start growing on that, if they have the LAC-Z gene, they'll pop blue. It's a really easy, just with the naked eye, you can see this ring of blue around the colony. And so that's a positive hit that, oh yeah, my biocontrol agent is still there. It's still persisting. Hopefully, that's what you see. You've probably heard of Lux genes for bioluminescence, maybe, or just the idea of bioluminescence, maybe. And that's another popular thing to do, and just have your colonies bioluminesce. So that's another strategy. So these are just easy, well, relatively easy visual ways to assess your samples. And then green fluorescent protein is another one that's really gained a lot of popularity, green fluorescent protein. So that gene that allows for them to fluoresce, they actually took it from a jellyfish, but you can put it into microbes and they'll maintain it and it'll work. So that's another strategy that you can do. And there are protocols for these that aren't too terrible in terms of getting them to work, but they can be pretty powerful. Here's some visuals of that. So here, figure A is a green fluorescent protein, and you can see it's really easy to spot. These are all a bunch of bacteria that are getting into roots of clover plants. So these are bacteria that hang out with clover and fix nitrogen. And you can see that even when they're in the root hair, you can see the bacteria with that green fluorescent protein. And then on the right too, I mean, look at that difference, the red background of the roots versus the green of the bacteria. So very nice contrast. The figure B is also nitrogen fixing bacteria that are gaining entrance into the plant via the root hairs. So they're going to get in, the plant has allowed them to come on in, and they're going to set up camp in the roots and start fixing bacteria or fixing nitrogen. So we'll talk about that a little bit when we get into plant bacteria interactions. So some good techniques out there. Sometimes you want to know how many dead things are in your sample versus living. And again, you can do that with epifluorescence. You can pick stains that are known that will only fluoresce if the bacteria or microbes in general are living and carrying out their physiology to stay alive, making their ATP, so respiration. And so that's not too terrible. You can do acridine orange and add in the INT stains, and that works. And so you can ask that question as well if you want to know, maybe there was a toxic spill and you're curious to know what did that do to the microbial population, and you can look at living versus dead. Here's another one that I don't particularly ever use. I think it's pretty fussy, but and I think it's a lot of people are saying the same thing. There's other better technology out there. This is called the direct viability count. So you take your sample and you incubate it in this chemical, nalidixic acid, and what it does is it doesn't allow for full cell division, but it allows the cells that are active to get to the stage where they elongate. So right when cells go through reproduction, so one bacteria, if it's like, okay, I'm gonna go through reproduction, what it does is it just enlarges, copies everything in its cell, including the DNA, and then splits in two. And so what this is doing essentially is arresting it in the elongation phase or the getting bigger phase. And then you have to have a fine enough visual that you can tell the difference between a plumped up, ready to reproduce cell and just a big microbe. So that can get a little tricky. And then slow growers may be missed because you didn't incubate them long enough and you just missed it. So there's issues with this one. I find that you have to really train your eye to this technique, and I'm not sure it's worth it. There's other techniques, but I just wanted to mention that that is something that they've done. It's a cool idea. It just is tricky visually to do. And then autoradiography is still being used. You can label with radioactive, and nowadays you can pick radioactive materials that aren't so dangerous to work with. When I was a grad student, I did some audio radiography and I was working with the really potent stuff where you had to wear the big lead aprons and the lead gloves, and yeah, that would, boy, talk about make me sweat. I've never sweated so much running an experiment because I was so nervous. And I had someone who's well trained in this technique helping me. Of course, she was like 6 feet back, giving me directions. So not something that's I would recommend, but there are less dangerous radioactive substances you can work with. Although having said that, our department has pretty much phased out all radioactive experiments. When I first got here, we were doing that routinely. Certain people were. And then in fact, I was the, what do you call it? I had a title. If you work with radioactive materials, you have to have someone go to those labs and use a Geiger counter and assess if there's been any spills or anything. So yeah, I got to do that because I was the new hire. So they're like, let Donna do it. So even though I didn't work with radioactive material, so you have to wear those badges that will soak up any radioactivity, you know, as you work, and then you have to send that in and keep your fingers crossed that it doesn't report back that you've been exposed. Yeah, that was fun. It was a lot of fun. So I would not recommend this route unless you absolutely have to. we have a lot of other technologies, but you see a lot of papers published, mainly older papers published.
What year did they start phasing that out?
Probably early 2000s was when they started phasing it out. So, which I'm happy. I was happy to give up that task because that was just, it makes you sweat and worry. So, all right. So here's viability staining results. So again, unless you're colorblind, which would be challenging because this is red green. But here you can see the live are green and the dead are red. And so this is a nice way to do it by picking the epifluorescence technique. I thought, that's, you just have to not have the red-green color blindness. You might have heard of this technique at some point. FISH, fluorescent in situ hybridization. When this first came out, it was a big deal and it was very powerful improvement in techniques. So this is a probe again, a DNA probe essentially, or RNA probe, but it has to be complementary to whatever target gene you're hunting for, or the messenger RNA that you're hunting for. And so this can really get you to answer several questions. You could look at phylogenetics, just who's there, separate them out by different species. They use it in the food industry when they're sampling food for contaminants. They'll often use fish. Clinical diagnostics, if a patient comes in sick, they will often turn to this method because it's fairly rapid. If they're concerned that there might be a certain pathogen that's causing the issue. So this is something that's been around for a while and is still used. I thought this was a nice contrast. So on the left, both of these are the same exact image under the microscope. On the left is what you would see without any staining, just looking at it normally under the microscope. And then on the right, they did the FISH technique. And you can see clearly that there's some different microbes showing up. You have the yellow and blue. They were focusing on the bigger cells. They noted all these big cells here. And they're like, I wonder what's going on here. Those are pretty big. So they ran the FISH technique and you can see that they were different. They look the same on figure A. The big ones all, oh yeah, they look the same, but they're not. And so again, getting at more detailed analysis of who's there in this case. They were wanting to know how much diversity is there. Again, really bright, colorful images, sewage sludge. You can imagine there's a lot of microbes in sewage sludge. So they were able to tag different genes and look at what's going on in these different samples. So pretty powerful technique. In microbial ecology, this type of technique is heavily used. And you can see that they used a confocal. Figure B, they say confocal laser scanning. So we have one of those as well. So you can get some really nice images. One of my former grad students, he actually took this class and turned the project that he did, the research project in this class, into his master's thesis, where he was isolating yeast off of apples from different environments. And he was using the confocal and doing some nice staining. And he discovered they had yet to be discovered, 2 species of yeast that had never been discovered. And he got to discover them. And then if you discover something, you get to name it. So he was having fun with that. But that being able to image them, he had to get really nice detailed images of the yeast. So he was using confocal and did some staining with that. Cardfish is a technique that it's just a kind of a twist to the FISH technique. It's an improvement on this. And so you may see that in some of the papers that you come across, cardfish. It's just an improvement on the FISH technique. So. They have to come up with all these acronyms. Catalyzed reporter deposition card. So here they were using that technique. And again, it's just a technique that you can turn to. In this case, they were hunting for archaea. They wanted to know how many archaea were there, and so they were hunting for that. So a nice technique. This table is, the columns are several different options. So here you have colony forming units in the first column. So this is a water sample. So colony forming units, that's the viable plate count. You're counting colonies. So they did a dilution, spread plate, incubate on, those plates, and then counted. And then they direct counts means that they did microscopy of some sort. Total biomass, you can actually get biomass numbers as well using different techniques. It's tricky with bacteria, but it's more straightforward with fungi. Active bacteria, so in this case, they used autoradiography to get at respiration. We're taking certain genes to look for activity of just that they're alive and kicking. And then they did a glucose uptake experiment. So I just put this here to show you that depending on the technique you're using, look at the data varies. And so stating what technique you used is really important, and thinking about what technique to use is something you have to think of before you actually go and do your experiments. So quite a bit of difference in the data. But you can see you can approach your question from many different methods, angles. And it just depends on, a lot of it depends on your resources. How much money do you have and time? But that's why you're always super detailed when you write up your methods section, whether it be for a research paper or your thesis or a publication. You have to be very detailed because it really matters how you process your samples and then how you test your samples. So that viable count, I just want to emphasize that you're definitely underestimating what's really there in your sample. So if it's a soil sample, for example, not everyone in that soil likes tryptic soy agar. And so you're only going to get a fraction showing up on those plates. And again, it's very selective. It's only those microbes that like tryptic soy agar that are going to grow. And if you want to get around that, like I said, you're going to have to make like 10 different recipes, which gets super expensive. And so, and of course, only the living microbes grow because it's a viable counting. You're only counting those that made it through the incubation period and showed up as colonies. Another viable count method is called the most probable number, and this is often done in clinical settings. It's a little bit quicker than a viable plate count where you're working with agar plates. A most probable number, you can do it a couple of different ways. And I'll show you a probable count in a second here. I didn't teach you the pour plate technique. We didn't do that, and that's kind of getting phased out because the spread plate's actually a better method. But an older method used to be that you would have agar in a test tube that is liquid still, and then you'd have your sample and you'd mix it into that agar, and then you'd pour that into an empty petri dish and let it solidify. And when you do that, you get microbes growing in the agar, microbes growing on the agar. And that used to be a thing where they were looking at, can these microbes grow in the agar? If you grow in the agar, you're capable of dealing with low oxygen levels because the agar doesn't allow for oxygen to diffuse. So it was a kind of a way to get at that. But now there's better ways to ask that question. But you may still see that in some of the older literature. So I just wanted to point out that that used to be a thing. So this is the most probable number, and it's broth instead of agar plates. And what you do is you take your sample and you serially dilute it. And then you incubate all of those. And then you look for turbidity. We use the word turbidity, which means the broth gets cloudy. And you go to the very last dilution, okay, so you see, oh, okay, at 10⁻⁶, there's no dilution, but at 10⁻⁵, there is. So they say the last tube showing growth, 10⁻⁵. The rule of thumb is they've just all decided this, and it works out, it's pretty accurate. The last tube showing growth, they say that growth developed from 10 or fewer cells. So that's your number, and then you can figure out what your population, your most probable number is in terms of population sizes. So you can calculate that. So this is a little quicker than agar plate method. They grow faster and you can get a little quicker results, especially in clinical settings. And then you've already done the spread plate in lab. Here they're doing the pour plate, bottom. So you did the spread plate up top, the pour plate's on the bottom. And then counting colonies allows you to then estimate, as long as you know what dilution that plate came from, you can count the colonies and then multiply by the dilution factor to get the estimate of what's in your original sample. And so with the spread plate, so when you're doing your research, you might want to know like how many microbes are in this sample. The spread plate would be a way to go. And then Duncan can show you how to do total estimates, total population estimates on that. Here's a fun one. Colony hybridization. This is an older method, but it's still a nice one to still use under certain conditions. So you get your colonies on a plate. So you do the spread plate technique, get your plate, you have all those lovely colonies. And what you do is you have this membrane that you just very gently lay down on top of your colonies. So they call it a hybridization membrane. So you just lay it down really, really gently and then really carefully peel it off your plate. And then you process that membrane. And so you take that membrane, so now it has all those colonies dotted on the membrane. Keep your spread plate, save that, you need it. But take that membrane and run your hybridization protocol. And so maybe you're asking, maybe you took a soil sample from a site contaminated with benzene. And so you ask the question, how many of the microbes that I've isolated have the gene to break down benzene? So then you transfer it to the membrane, you have your probe looking for the gene that breaks down benzene, and you mix that into your assay as you're working with the membrane. And then you look to see what pops, which colonies are popping for that. And so this is the flow chart for this idea. And again, when you finish it, hopefully you'll find some of the colonies. Oh yeah, they have the gene necessary to break down benzene. Then you can go back to your spread plate, pick those colonies and start working with them. Like maybe you want to grow them up to high numbers and start spiking them into the system that you got your sample from to help break down benzene. So it's a nice, pretty quick method once you have your spread plate. So that's kind of a fun one to work with. Here you're seeing the results of a colony hybridization dot blot. DNA hybridizations. So here they were just looking for Burkholderia cepacia. That was their question. Is Burkholderia present in this sample? And then really dark colonies, that's yes, that's Burkholderia. So this is an older method, but still really can be very useful. Okay. So I wanted to give you a couple of examples from the literature on scientists who have used some of these methods to kind of put it into context, I guess. So this was published back in 2020. Soil microbial community structure is affected by biochar and fertilizer sources. Do you know what biochar is? That's not a terribly well-known thing. Biochar can be made from different substances, often from wood, like tree trunks. And you essentially process the tree trunks, or they've done it with coconut husks as well, where you take the material and you burn it in an anaerobic method. And it creates this charred wood or charred coconut husks. And then they grind that up into particle sizes, kind of similar to particles of sand. And then you can mix that into your soil. It's one of the really good advantages of biochar being mixed into your soil is that we're finding that it creates kind of a nice structural home, for lack of a better word, a structural home for the microbes in the soil. They love it. They love attaching to it and hanging out with it. And so it creates really good texture to the soil. So one of my grad students worked with biochar for some of his treatments. He was trying to grow different plant crops in very degraded soil and asking what can we do to this degraded soil to allow for good growth of these plants. This is primarily driven by the idea that this was when Detroit was very heavily involved in greening the city and that was like the start of it. They wanted people to start growing their own food and having, plots of land in neighborhoods turned over to growing food. And a lot of that land is degraded because it's primarily land that had houses on it, lawns, not necessarily well maintained for agricultural purposes. And so we were asking, like, he really wanted to know, if you have degraded soil, what can we do? And biochar actually was one of his best treatments. We had some really nice growth. It really helped improve the microbial populations in the soil too. So biochar is a big thing right now. It's more and more people are finding it very useful. So they were looking at this idea of organic fertilizer, right? Is it's going to let me draw on it. Or organic amendments can potentially mitigate the deleterious environmental impacts of inorganic fertilizers. So if you over fertilize with inorganic fertilizers, like a lot of farmers, at least historically, and maybe not so much anymore, but historically it's like more is better. So they just throw a lot of inorganics because it was fairly inexpensive. Too much inorganic fertilizers can actually cause the microbes in the soil to crash. They don't do well under those conditions, and it actually can create a lot of death to the microbes. And so you can tell that they're aware of that. So they're thinking about the idea of organic amendments. But then, how does this impact the microbial populations? So they were looking at different sources of fertilizer and biochar.
So you're saying that you need to nutrient dense?
Yeah, the inorganic properties of the fertilizer, they actually called it a microbial desert. If you look at soil that has been over fertilized for extensive time periods, and you look at the microbial populations compared to like an organic treated soil system, it's very large difference in micro population. So, and that was not something they anticipated, obviously. When they came up with inorganic fertilizers, they thought it was the best thing since sliced bread, but there was some unintended consequences to that. So they were looking at poultry waste for organic fertilizers, as well as inorganic, and biochar as well, on chili peppers. So they did inorganic nitrogen at 0, 50, 75, and 100%. And then they had organic nitrogen amendments too, and they were looking at mycorrhizal fungi. So arbuscular mycorrhizal fungi, it's a type of mycorrhizal fungi. And then they looked at gram-positive bacteria, gram-negative bacteria. They were looking at phospholipid fatty acids. Right, so some of these things that we've been talking about in this section of the class they were running that. So here they're just looking at phospholipid fatty acid profiles. And the no fertilizer is the far left bar, and then the control was all just inorganic. And then bio means the biochar addition at different volumes, and then organic nitrogen as well, adding in. And so you can see that, especially at 30 and 50% biochar, and then with the organic nitrogen, you can see that it did do better in terms of really increasing the phospholipid fatty acid profile, which usually means you can equate that with your microbial populations are going up. You're increasing the microbes in the soil. Here's another set of data that they were collecting. So they were able to look at with the phospholipid fatty acid profile, you can use that to detect gram-positive bacteria versus gram-negative. That's what you see on the top two bar graphs. And then you can also take for fungi with the phospholipid fatty acid technique. You can specifically look at fungi, and then they can look at mycorrhizal fungi in particular. And then they are even looking at actinomycetes, which Streptomyces bacteria that I work with, they fall into that group. And then they were looking at eukaryotes, or just saying how many eukaryotes are here. So this, I put this up here to try to show you the real world application of that technique we talked about with phospholipid fatty acids. And that you can get quite a bit of data from that. And then they were looking at the same thing. And then here they were looking at fresh pepper yield. Two different growing seasons. So the bar graphs are the two different growing seasons. And so this type of method for what they were asking was generating some really good data that they could then think about and draw some conclusions. So I'm not asking you to like read this paper and memorize what they did. That's not the point. I'm just trying to get you to think about how can we apply some of these techniques, like what kinds of questions might it help answer as you are learning about these methods. And then here, this is a fertilization experiment that they did. They said what kinds of fertilization and how they did it had a greater effect on the soil microbes than if they did a crop rotation process, or they looked at what growth stage the plant was in, because sometimes that can impact the microbes too. Is what, is the plant really young? Is it really old? Is it busy making fruit? That kind of thing. So they were looking at several things, and this is the experiment, 33 years. This was a 33-year-old experiment. That's pretty phenomenal that they had the ability to carry that out for so long. And so they were asking, what's going on with the microbial communities under these different treatments. They looked at bacteria and fungi at when the plant was growing, flowering, getting to maturity, winter wheat and soybean. Again, no fertilizer, only inorganic. NPK means inorganic. And then you had wheat straw and cow manure as your organic. And so they had, inorganic and organic. And then they were using ribosomal RNA techniques, which is super valuable. ITS, amplicon. So these were ways of DNA sequencing and looking for, ITS looks for fungi. If you use that process, that's specific for fungi. And then 16S ribosomal RNA, that's specific for bacteria. So again, this idea of using these techniques to answer some of these questions, this is real world application. And again, it looked like the organic had a positive effect on most groups, but not all. Okay. Okay, we'll stop there because the next one I want to show you takes a little bit more explanation than a minute. So we'll end there. And we'll be wrapping this topic up and starting into